Good morning, everybody. So my talk today is going to be sort of.
Sorry.
Well, now everyone's awake. My talk this morning is going to be sort of a general overview to this algae pollution problem, and I look forward to my colleagues, Dr. Rosen and Parsons' talks later, which I'm sure will give a little more detail on what's going on. But I just wanted to share some observations and general thoughts about algae as an indicator of nutrient pollution, specifically in the Caloosahatchee estuary. And I'm presenting from home today, so I apologize for any potential dog interruptions. Hopefully it'll be not too extreme. The first point that I'd like to make is that algae is a short word that describes a very broad and complex group of organisms. Basically, any photosynthetic organism other than a true plant, a true vascular plant like a seagrass, would be classified as algae. So that's everything from the simple prokaryotic organisms like the Cyanobacteria, which are photosynthetic, to more complex eukaryotic algae like diatoms, and then even multicellular algae, which we call macroalgae or seaweeds. I may use some terms synonymously here. If I say macroalgae, I mean seaweeds, the multicellular algae. If I say microalgae, that could mean the microalgae that drift in the water, which we call phytoplankton, but it could also mean microalgae attached to the bottom or attached to seagrass blades, which we can refer to as algal scum or epiphytes. Next slide, please. So is algae good or bad? It really depends. The algae does many beneficial things for the ecosystem. It's a photosynthetic organism, which of course creates oxygen, absorbs carbon dioxide, and absorbs nutrients. They're the base of most aquatic food chains, so really, really important for creating the primary productivity, which passes up the food chain to smaller grazing organisms and then on to commercially important species. Algae can provide habitat as well, particularly multicellular algae like seaweeds provide some ecosystem services that are similar to seagrasses in terms of providing habitat. And the algae themselves are a diverse group which contributes to biodiversity, and also because of the food and habitat roles, they contribute to biodiversity. So that there's a lot of good in algae, but of course we know well that algae can have many bad effects as well. They can make the water murky and stinky. Some algae produce toxins. Algae can compete with other organisms in marine ecosystems like corals and seagrasses and can smother and kill those other organisms. And when we get large blooms of algae, they can decay and cause problems with low oxygen, which we call hypoxia and anoxia. And ironically, even though algae in a good scenario can increase biodiversity, when algae are overpopulated and causing these other problems, they can reduce biodiversity and reduce the structural complexity and productivity of ecosystems. Next click. And so one of the things that shifts algae from good to bad is nutrient pollution, the excessive loading of the environment with nutrients, which of course is one of the things that the CHNEP is focused on reducing. Next click. So as an example of how nutrient pollution can lead algae to become a problem, I'm focused on my personal favorite ecosystem, seagrasses. And this is something the public particularly can get confused about because, of course, seagrasses and algae both require nutrients to live, and yet an excess introduction of nutrients to the environment causes a shift from the seagrasses to algal-dominated systems. And that occurs because although both seagrasses and algae need light and nutrients, the algae have lower light requirements and are more able to take advantage of a high amount of nutrients. So when nutrients are in excess, the algae become overabundant, they block the light, and the seagrasses decline. And so the two groups of algae that can cause this harm are the microscopic algae suspended in the water, which we call phytoplankton, but also the algae on the bottom, which are called benthic algae. And that can include both seaweeds and benthic microalgae, the algal scum or epiphytes that grow directly on the bottom and the seagrass blades. Next slide, please. So we can really think of this nutrient loading thing as kind of a spectrum. And this is a conceptual ecosystem model from the University of Maryland, which I like to use because it shows the spectrum moving from left to right of the type of ecosystems we would expect in shallow coastal waters as you go from a very nutrient poor environment to an environment that's over enriched with nutrients. And of course, here in Southwest Florida, we're well over on the right-hand side of the spectrum. We really don't deal with a lack of nutrients here. We're sort of in the area where we have an excess of nutrients leading to the replacement of seagrasses with macroalgae, seaweeds, phytoplankton, and benthic microalgae. And so they call this process eutrophication, rhymes with nutrification, and it's sort of the shift from a seagrass dominated system with clear water to a system that's really overwhelmed with algae and phytoplankton. Next slide, please. Next click. So this is sort of the simplified view of the diagram, but of course there are a lot of things that can complicate this scenario. Exactly how eutrophication plays out really depends on things like the depth of the water. In deep water coastal ecosystems like Tampa Bay, the primary algae that they have to worry about as it becomes more nutrient enriched is phytoplankton darkening the water. Whereas here in Southwest Florida, where our estuaries tend to be more shallow, we have to worry a lot about the benthic macroalgae, and we need to consider that and the phytoplankton in our assessments. Another thing to think about is that nutrients are one of the factors that can lead to this overgrowth of algae, but there are other aspects of the environment that can contribute to algae growth as well, such as the amount and type of grazers in the environment. Grazers can be beneficial by removing algae from the seagrass, or grazers in some cases could just contribute to the stress by eating the seagrass as well as the algae. Filter feeders can be very important. Organisms like oysters and clams can remove some of the phytoplankton from the water and help supply light to the sea bottom. And then another thing to think about is that there are hundreds of species of algae, if not thousands, in Florida. And not every species is equal. Some are benign or beneficial, and others, like infamously red tide and Microcystus, cyanobacterial blooms are very, very harmful. And different factors of the environment, such as salinity and light levels, and nutrient ratios, like the ratio between nitrogen and phosphorus, can determine what kind of algae it is that's blooming and how bad the effects are. Next slide, please. So I mentioned that there are hundreds of different kinds of algae in Florida, and many of these types of algae can become a nuisance. And so when an algae bloom is a problem, we call it a harmful algae bloom, abbreviated HAB. And this is a slide that I presented at a real estate investment association in 2018 when we were experiencing a lot of harmful algal blooms at the same time in Florida. And so this is sort of a rogues gallery of some of the harmful algal blooms to watch out for in Florida. The first on the left-hand side here is the type of algal bloom that affects Lake Okeechobee, freshwater canals in Cape Coral, and the Caloosahatchee Estuary when there's a lot of freshwater flow. This is Microcystus eruginosa cyanobacterial bloom, which I'm sure you'll hear more about from Dr. Rosen later. The problem with this species is that it's toxic. It kills aquatic life, and it also shares one of the problems that all of these harmful algal blooms have in common is that the sheer biomass of algae, even if the algae is not toxic, can create problems with darkening the water, creating excessive demand for oxygen and low oxygen conditions. And the principal culprit behind the excessive abundance of this first type of algal bloom, the Microcystus eruginosa cyanobacterial blooms, is of course pollution, specifically nutrient pollution. The next villain is Florida red tide, caused by dinoflagellates in the genus Karenia. The problem with this is that, of course, it's toxic and it kills marine life. And the causes of this are complex, and they include some ocean weather conditions, upwelling and winds, but also anthropogenic pollution from land-based discharges, as the committee mentioned recently. have been reviewed by scientists like Miles Medina. The consensus is that anthropogenic nutrient loading exacerbates this algal bloom as well. Another algal bloom that doesn't affect our area so much as the East Coast is this brown macroalgal bloom, sargassum algae, and that one's sort of really a global scale problem caused by sort of nutrient enrichment of the entire ocean basin from multiple sources, everything from the Amazon to African dust. And then when weather conditions concentrate the algae, it can be a nuisance along the shorelines. Along our shorelines in Southwest Florida, most of the macroalgae seaweed that washes up are various species of red macroalgae. So there's dozens of species of this stuff, and it's more of a nuisance than a major threat. But when this red macroalgae is overabundant in our coastal estuaries, it can be a real problem because it outcompetes and can smother seagrasses. And of course, this species responds to nutrient enrichment. So its principal cause is pollution as well. Next slide, please. And the list of obnoxious algal blooms grows. One of the types that we see sometimes in the offshore environment here is a type of cyanobacteria entirely different from the Microcystus in the freshwater. It lives in a completely different type of environment. But one of the interesting things about this Cyanobacteria trichodesmium is that it is not particularly harmful or toxic in itself, but because it's a nitrogen fixing organism, it can help fuel red tides. And then any factors that therefore stimulate this cyanobacteria could indirectly be stimulating red tide. So scientists are concerned about loading of phosphorus and iron into the ocean through atmospheric deposition and other sources, overstimulating the trichodesmium, which in turn will overstimulate the red tide, which is sort of an example of the complicated chain reactions you can get where one type of algal bloom eventually begets another type of algal bloom. There are some other types of algal blooms that we have in estuarine environments. One particularly sinister kind in the estuaries on the east coast of Florida, which could also affect our estuaries, is something else that they call brown tide, caused by a phytoplankton called Oreoumbra. And this one's not directly toxic, but it blooms in such densities that it makes the water very, very dark and contributes to low dissolved oxygen problems. And this is one of the main species that was part of the so-called super bloom in the Indian River Lagoon that ended up wiping out massive amounts of seagrass there and contributing to the tragic manatee starvation that we've seen in the last year or so. Moving on, we have this type of filamentous cyanobacterial bloom that forms on the bottom of our estuaries called Lingbia. Lingbia is the old scientific name for this thing, which has now been reclassified as Dapis bluosa. I'm sure Dr. Rosen could give you more information on the classification of this. But this is a threat both because it smothers seagrasses. This is actually a seagrass bed that I took a picture of in Estero Bay, which has just been totally covered with this brown carpet of Lingbia filamentous cyanobacteria. And in addition to the sort of light blocking seagrass smothering effect of this, it is somewhat toxic and can disrupt the food chain in estuaries. The final species, I promise this is the last species of algae I'll talk about specifically, is Caulerpa fastigiata. There are many species of algae in the genus Caulerpa. Some of them are more associated with clear water reef environments, and others are associated with dirty water estuarine environments. This Caulerpa fastigiata flourishes in dirty estuarine environments where there's an excess abundance of nutrients, particularly nutrient loading from the sediment seems to stimulate the abundance of this green algae, which has become a real problem in parts of Charlotte Harbor and the Caloosahatchee estuary. This picture was taken by Rick Bartleson from the Sanibel Captiva Conservation Foundation in Matlacha Pass, where this species of algae just since 2018 has really taken over. Next slide, please. Right, so the Caloosahatchee River and estuary is our sort of focus of study. I'm work at Florida Gulf Coast University, and this is our sort of backyard estuary. And there are some water quantity and water quality issues with the Caloosahatchee estuary that I'm sure this group knows very well. We have water quality issues stemming from the local watershed and the usual suspects of wastewater and stormwater problems, and sort of poorly controlled nutrient pollution from the local watershed are an issue here. But we also have this infamous issue of outside inputs to the estuary from the synthetic link to Lake Okeechobee, which has sort of expanded the watershed to this much larger area where when water is released from Lake Okeechobee into this estuary, we get these massive changes in salinity and also a lot of extra nutrient loading. Next slide, please. Some of the harmful algal blooms that we've had in the Caloosahatchee estuary and associated Southwest Florida waters in association with major water releases, which are sort of a combination of weather events and the water management changes that we have here, include Microcystis eruginosa blooms in the low salinity areas. And you can see this picture of sort of a green scum carpet covering a bunch of dead fish on the shoreline. This is a picture that I actually took out in Sanibel in 2018 when there were very high water flows, and this freshwater algae was actually making it all the way out into the Pine Island Sound around Sanibel. And it, of course, wouldn't survive there because the salinity was too high, but there were such large amounts that it was piling up along with the dead fish, which were actually killed by the algal bloom that you see identified in the FWC map on the right, which was a red tide bloom. So this was kind of like the summer of really bad conditions in the Caloosahatchee estuary, where we had this freshwater cyanobacterial bloom that was blasting out into the Gulf and meeting the nasty red tide bloom, and it was just really a disgusting soup of death that was unpleasant to work in. Next slide, please. So partly stimulated by the problems that we had with this series of algae blooms in 2017 to 2020, FGCU received some funding to monitor some of the benthic habitat indicators and water quality in the Caloosahatchee estuary. So there's a history of using benthic habitat indicators like the abundance of seagrasses and oysters in the estuary as a metric of water quality. And we continued this and tried to integrate and update some of our update the historic maps, which oddly the last time we had like a really comprehensive picture of seagrass coverage in the Caloosahatchee estuary was from 1993. So we did a estuary-wide assessment of what was on the bottom, and although our main objective was to quantify the seagrasses, the SAV or submerged aquatic vegetation, we also took inventory of macroalgae, what types of algae were growing there, and we looked at other benthic habitats as well, and we looked at the salinity levels and the water and the optical water quality. Next slide, please. The way we organized this study was by dividing the estuary up into different salinity zones, zone A through zone E, with zone A being the lowest salinity zone where we usually find freshwater life, and zone E being the San Carlos Bay region where there's a lot of exchange with the Gulf of Mexico, high salinity and a high diversity of marine organisms. We covered the entire shoreline here with transects every kilometer, so 100 shoreline perpendicular transects. And the next slide will show some of our water quality from these transects, and then we'll look at some of the benthic life from these transects. Next slide, please. This sort of just gives you an idea of the salinity and water quality conditions along this estuarine spectrum from the Gulf to I-75. And the salinity diagram is color-coded with blue indicating the marine salinities and red indicating the freshwater salinities. And then there's this long transition zone where the salinity is neither here nor there. As far as water clarity is concerned, it was at the time that these data were taken, it was much more clear in the San Carlos Bay near the Gulf of Mexico, and much murkier further up the estuary. These data are KD, which is a light attenuation coefficient that we determine using paired optical water quality sensors. and this knowing the light attenuation coefficient tells us basically how much light is being blocked as it passes through the water, and we can use that data to figure out or predict what depth seagrasses and other underwater plants would be able to live at and receive enough light. So obviously, where the water is murkier, seagrass is going to be restricted only to the very shallow areas, whereas with water more clear, the seagrass can inhabit deeper parts of the estuary. Next slide, please. This is our sort of habitats map that I was really excited to see all of the different things that live on the bottom of this estuary, despite the harmful impacts that it's experienced for years. We do have a diversity of seagrasses in the estuary, and the first 5 species listed here are the 5 are seagrasses. Syringodium filiforme is manatee grass, found only in the highest salinity regions of the estuary. Thalassia testudinum is turtle grass, also restricted to the high salinity areas. The 2 species of Holophila decipiens and Engelmannii are small seagrasses, which tend to be mixed in with the other seagrasses. And again, they're found in the high salinity parts of the estuary. And moving further up the estuary, the only seagrass still present is Halidule wrightii, the shoal grass. And further still into the estuary, there are a few seagrasses. There's a lot of hardened shorelines in this part of the estuary and not much habitat for seagrasses. Plus the salinity is quite variable, which is hard for seagrasses to tolerate. But there's a lot of oyster beds in this part of the estuary, and that's the sort of light beige colored dots there. Further up into the estuary, there's one, I suppose you could call it a seagrass, but it can live in fresh water as well. It's Rupia maritima, which is sort of the most prevalent species of seagrass in the middle and upper part of the estuary. And then there's this formerly abundant submerged aquatic vegetation, Vallisneria americana, which is a freshwater plant. And it does survive in the upper estuary, but it's nowhere near as abundant up there as it used to be. You used to have really thick SAV beds in the upper estuary formed of Vallisneria americana, which were sort of comparable with the thick turtle grass beds that we now have in the lower part of the estuary. All right, next slide, please. And this is just sort of describing some of the changes that we saw when we compared the current underwater state of the estuary to the last map that was made in 1993. We've really seen this major contraction in the range of the Vallisneria americana, the tape grass in the upper estuary, and we've seen a decline in some of the seagrasses in the lower estuary as well. Next slide. One thing that was, of course, flourishing in the estuary, though, was macroalgae, so seaweeds. And these red dots just show macroalgae in general, so this doesn't break it up by species. But you can see that there was a lot of macroalgae all throughout the estuary. And there was a surprisingly large amount in the upper estuary in the low salinity regions. And with the dominant species there was Polysiphonia subtilissima, a type of red algae. Rick Bartleson from SCCF helped us identify this algae. Further down in the estuary, there was a lot of algae as well, but it was made up by a variety of different species. And in fact, only in the most seaward part of the estuary did seagrasses outnumber algae in terms of abundance. So we'll look at the next slide. This graph shows the 5 zones of the estuary, zone A being the low salinity zone and zone E being the high salinity zone. And the green bars are the amount of seagrass and the orange bars are the amount of algae. So you can see in every part of the estuary except the saltiest part of the estuary, algae overshadows seagrass. It's much more abundant than seagrass. It's a really stark contrast in the upper estuary where there's this very high cover of filamentous Polysiphonia subtilissima and just a tiny amount of seagrass. Another thing that you can see is that the seagrass is more light sensitive or has higher light requirements than the algae. So the seagrass tends to be restricted to the shallower depths, whereas the macroalgae is still able to be abundant a bit deeper, which is one of the things that gives it an advantage as water quality deteriorates. So where there is seagrass, it's most abundant in the shallowest waters, whereas the macroalgae is more abundant a bit deeper down. Next click. One interesting thing that we found in the middle part of the estuary was that there was little seagrass in the very shallow areas. And this is the part of the estuary where there's a lot of hardened shorelines, a lot of seawalls, and there's just not that much good shallow water habitat for SAV in that part of the estuary. Next slide. This slide here gives sort of an overview of the species composition, the types of algae that we found in the different zones of the estuary. So I've just listed them out with their scientific names and color-coded the scientific names by what type of algae we found there. So in the upper estuary, that red filamentous algae, Polysiphonia subtilissima, was the dominant type, but there were also a lot of benthic diatoms in the genus Melocyra, and there was some filamentous cyanobacteria. This is something that we identified with a microscope, but also my colleague Toshi Urakawa did a microbial analysis of epiphyte algae samples from all the zones of the estuary, and he was able to identify a high abundance of cyanobacteria in the epiphyte communities of different parts of the estuary there. Further down in the estuary, you start to get a higher diversity of red algal species as some of the marine algae show up. And by the time you're in zone E, the most seaward part of the estuary, there's a very high diversity, not only of red algae, but of brown algae and green algae as well. One of the types of algae that's found in the sort of moderate salinity part of the estuary, zone D, is Caulerpa fastigiata. That's the one I showed a picture of earlier, which is the green carpet. And I'm going to say a little bit more about this Caulerpa fastigiata in a couple of the next slides, because it's an example of how the algae can harm the estuary. Next slide. So this is a mat of Caulerpa fastigiata in Matlacha Pass. You can see a tiny amount of Halidule wrightii seagrass in the left-hand part of this picture, but it just looks like a thick shag carpet of Caulerpa fastigiata. And this is not an isolated patch, but this is something that has in recent years come to cover the entire bottom of the estuary in Matlacha Pass, which of course is attached to the Caloosahatchee estuary. Next slide, please. This Caulerpa fastigiata is a native species, but it can be quite a nuisance because it sort of flourishes in these nutrient-enriched areas and outcompetes seagrasses and causes environmental problems in the estuary. It overgrows everything. It reduces faunal diversity because there are some species that will eat it, but relatively few species besides one type of sea slug will actually eat this thing. Next slide, please. And this, I mentioned it had become incredibly abundant in a few years. This is a picture from Ralph Arwood's aerial photography. And it looks like the entire estuary here has got this dark covering on the bottom. And if you didn't know better, you might think that this was a thick, healthy seagrass bed, but it turns out that this is actually just almost a monoculture of this one benthic algae, which has almost entirely replaced the seagrass. Next slide, please. There are some grazers, this one species of sea slug, Oxynoiantillarum, which can eat this stuff, and it, of course, becomes abundant in the areas where the algae is, but it just can't keep up with the rapid growth of the algae. Dr. Bartleson has monitored the nutrient concentration in the ground pore water immediately beneath the algae and found that it's getting this extremely high supply of nutrients from decomposing material in the sediments beneath these algal mats. So even though the waters above the algal mat have relatively modest amounts of nutrients, the sort of legacy nutrients that are leaching out of the sediment, possibly coming from groundwater sources as well, are fueling this stuff from below and helping it maintain these super high abundances. The really thick algal mats really have no enemy except themselves. Next slide. And when the water is warm in the summertime, the algae's biological oxygen demand at night creates these low oxygen conditions that create hypoxia throughout this part of the estuary. The algae dies, and this massive release of hydrogen sulfide into the water from the decomposing algae then interacts with other chemicals in the water and creates this precipitation of white sulfur, discoloring the water like it was coffee with milk in it or something. So it's obviously this really harmful condition that we see, and it seems to be sort of spreading throughout the estuaries of the region. Next slide, please. And this has been happening year after year, so it wasn't like a one-time freak thing. It did seem to sort of start in the aftermath of the major 2017's Hurricane Irma, but now it seems to be sort of feeding into itself, and I'm concerned that this is going to be sort of a chronic condition for the estuary. Next slide. And this is a major concern because of the biochemistry associated with the growth and decay of this algae. When you have this massive amounts of decomposing material on the bottom, the sulfur cycle is overstimulated. You get these microbes which use sulfate as an electron acceptor in the oxygen poor environment under the algal mats. That creates hydrogen sulfide, which is a toxic compound. In some areas where there's a high abundance of this decomposing algae and the bottom is disturbed by boat wake, for example, you can actually have a huge amount of hydrogen sulfide gas mist up into the air, and it could potentially kill a person. Where there are similar macroalgal blooms in other parts of the world, like France, people have actually died if they've gotten sort of caught in one of the clouds of hydrogen sulfide from one of these decomposing algal mats. Next slide, please. So in summary, the problem that we're seeing here is in all parts of the estuary, a really high abundance of benthic macroalgae, oftentimes greatly exceeding the abundance of the beneficial seagrasses. And this overabundance of algae is leading to biogeochemical dysfunction, which means like low oxygen conditions, sulfide release, and all kind of problems for humans and the environment. So the solution, as I'm sure you know well, is we need to reduce nutrient loading. But the severity of this problem, is indicated by all of these algal trends, indicates that we really, really need to work hard to reduce nutrient loading by a lot as quick as we can. And that means better wastewater management and stormwater management. So of course, it's really encouraging to see all of the stormwater and watershed enhancement projects that this group is working on. Thank you. I'd just like to acknowledge the funding that supported this research. My co-PIs at FGCU, marine chemist, Dr. Puspa Adhikari, microbial ecologist Hidetoshi Urakawa, my frequent collaborator at SCCF, Dr. Rick Bartleson, and all of the students, of course, that have carried out the research. Thank you.